So in previous classes we have already discussed about the genetic materials and the structure of DNA and RNA and the search for genetic material, the what kind of experiment take place for the discovery of genetic material. And now we will see the certain processes that take place in all the living cells. And we know that DNA is discovered by the Watson and Crick in 1953. When they discovered this double helical structure of DNA, they also suggested that this DNA goes to the RNA and protein, ultimately to the protein, which is by this way DNA express themselves in a cell. So, and they given this theory name is central dogma of molecular biology. So, what is central dogma of molecular biology? We have already seen that DNA makes copies of themselves known as DNA replication. So, this process is replication. Then DNA makes RNA. This process known as transcription. And then RNA makes protein. This process known as translation. So my dear students, today we are going to discuss about all these things, that how DNA makes copies of themselves, how DNA makes RNA, and how RNA makes protein. So first of all we will discuss about DNA replication. So let us start today's class. DNA replication. So what is DNA replication? DNA replication means the process by which DNA makes copies of themselves, known as DNA replication. And who suggested this theory? That the Watson and Crick, when they proposed the double helical structure of DNA, they also proposed that, they said that when we separate this double helical structure of DNA, we get two strands. This one is first strand and this one is second strand. And this second strand, this both strand, after some time goes into the produce another complementary strand. So Watson and Crick said, when we separate these DNA molecules into two strands, they both strand produce or makes their complementary copies and form a double helical structure again. So they said that the complete DNA is not work as a template. Here in DNA, for making of copies, the single strand is work as a template or original copies, by which here new base pairs will form, and again double helical structure of DNA will form. They said that both the strand, newly formed strand, having one parental DNA strand and one newly synthesized strand. So this method of DNA production or DNA making copies of DNA, known as semi-conservative mode of DNA. What is the meaning of semi-conservative mode of DNA? Which means the DNA conserve themselves semily, means only one strand having parental DNA and another strand is newly synthesized strand. Here you can see that the parent conserve their DNA in both the strand, send it to the both the strand, and both the strand produce newly synthesized strands. So this is known as semi-conservative methods of DNA replication. Now questions come arise that what is the evidence that DNA copies themselves in a semi-conservative mode. So two scientists comes up with their experiment, and name of the scientist is Melanson and Stars. They conducted the experiment on the E. coli for the evidence of semi-conservative mode of DNA replication. What they do? They take DNA, they take E. coli and grow them into the media containing nitrogen salt with radioactive 15N. This 15N is slightly heavier than 14N, which means when they grow in a 15N strand, 15N with a culture media, both the strands will have heavier nitrogen, and when they again grow them into the media containing 14N, so what will happen? They found that when they cultured E. coli in a 15N and centrifuge them, they found that DNA is present, that DNA have higher density. And after that, when they culture this E. coli in 14N nitrogen, they found that after culture in 14N, and then they calculated or estimated their measured the density of this molecule. They found that here 14 nitrogen containing DNA, and here 15 nitrogen containing DNA. But after one generation, they found that the DNA having intermediate density, neither lesser or neither heavier. They found that DNA is in the first generation having intermediate density. Okay, so this is indicate that both the strand will grow, which is having one strand of nitrogen, lesser density having 14 nitrogen, and one strand having nitrogen containing 14 and heavier nitrogen. So this experiment indicate that or determine that the DNA which is replicate is occur in a semi-conservative mode. Now actually what is the replication and what is this mechanism of replication? Now we will discuss about the mechanism of DNA replication, in which how DNA makes copies of themselves. We know that the DNA replicates and makes copies of themselves, and we also know what is the semi-conservative mode of replication. Now we know that only single strand of DNA work as a template. Means consider that this is the 3' end and this is the 5' end. Why this is called 5' end? Because the side phosphate group present on a 5' end, where OH group, hydroxyl group is present on a 3' end. Now let me give them a particular nitrogen base. So here phosphate A, phosphate T, phosphate C, phosphate G, and so on. Here phosphate G and phosphate G, here phosphate, and here OH group, because it is 3' end. Okay, so what will happen? The DNA which grow in 5' to 3' end, what it required? This is the DNA template, single stranded DNA template. How it will go? It requires particular primer for growing strand. Here this is the A, so the complementary base pair is T, P, T phosphate, P phosphate, T phosphate, A phosphate, G phosphate, here C phosphate, and so on. Now what will happen? This is the primer. Okay, it is very necessary for the production of new strand. The primer is very necessary. After the primer, one enzyme called DNA polymerase comes into play, and they polymerase the nucleotides. They add the nucleotides which is present in the nucleus. What will happen? They do the free nucleotide, which is A phosphate, triphosphate T, triphosphate C, triphosphate G. Okay, so these are the nucleotides present in a nucleus. Now what will do polymerase do? Polymerase take this nucleotide and bind here. Here look at here, the base pair is phosphate T. Okay, the polymerase take this nucleotide and bind it with the here phosphate A, phosphate A. Okay, so what will happen? And between them two hydrogen bond will form, and this phosphodiester bond will form between this strand, and this process is continuously grow and grow in a 5' to 3' end. Now question arise that why DNA polymerize the nucleotides in a only 5' to 3' end. This can be asked in exam, that DNA polymerase work in which direction? So DNA polymerase work in a 3' to 5' end direction. Now questions arise that why DNA polymerase only go in a 5' to 3 direction? So there is question. This shows the answer here, because the new nucleotides comes with the energy in form of phosphate. If it will grow from the 3' to end 5' end, what will happen? Here it comes up with hydroxyl group, and here phosphate A, it is a phosphate T. What will happen when this hydroxyl bind here? They will hydrolyze the phosphate group. As a result of this, and this hydroxyl group don't have energy, so it doesn't make any sense to provide the energy of new nucleotides from the strand. So here it indicates that life is smart and it doesn't go this way, it goes this way. Okay, so the polymerization take place, and this DNA polymerase continuously polymerize these DNA new strands like C, G, and then C and then A. Likewise, it will continuously grow. Okay, so this is the how DNA polymerase work. Now let us see in a DNA, double stranded DNA. This is 5' strand and this is 3, and here the lower strand is 3' and 2 5' strand. Here the one enzyme called helicase, they cut this strand, both the strand, into a single stranded DNA. Remember that the helicase name helicase. This enzyme also known as the molecular scissor. What this enzyme known as molecular scissor? That can be asked in exam, that which kind of enzyme known as a molecular scissor. So the helicase is known as molecular scissor because it cut the DNA into single stranded DNA. It converts single stranded DNA in double stranded DNA into single stranded DNA. Now after cutting, what will happen? The one primer will be added here. This is the primer. And question come arise that where from this primer come? So the primer comes from one enzyme. Hopefully that enzyme known as primase. Okay. After primase add the primer. And after primer, another enzyme is a DNA polymerase. DNA polymerase will continually polymerize this nucleotide, and new strand will continuously form from 5' to 3' end. Okay, this is 5' to 3' end. Everything is okay here. All right. But the questions arise in a second strand. Look at the second strand. As we know that DNA polymerase cannot work from 3' to 5' end. So here new DNA strand is having a 3' end. What will happen here? DNA polymerase work only in from 5' end to 3' end. So primer will bind here. Okay, so this is primer bind. Now what will happen? Another exam comes. It is a DNA polymerase. DNA polymerase bind here and continually bind. Okay, this is okay. But the questions come arise when, look, when this helicase continuously cut, and behind the helicase in a 3' to end, 5' to 3' end, continuously one strand grow here to here. But when on other hand primer bind here, okay, and then polymerase, then from here helicase cut this DNA. So what will happen? Again from here it will start here, another primer will add, and then polymerase, another primer, another polymerase. So another strand will grow in a fragmentation. So with the fragments. So this fragment is discovered by Okazaki.